Hey, good morning, Egan High School AP Bio peeps. What's up? Miss hanging out with you guys. I know what you're missing most though, daily trivia. So here we go. See if you can keep track here. Number one, the subject is Mexico today. What color are the three vertical stripes on the flag of Mexico? Hit pause. All right, you're back. The answer? Green, white, and red. Should be keeping track. That was our sample question. All right, freshman level. Number one. What is the basic unit of currency in Mexico? Easy one. You're right, it's a peso. Number two, which city is the most visited border city of Mexico? I actually have been there myself. Yes, it is Tijuana. Number three, freshman level question. Name a country that borders Mexico to the southeast. Southeast. There are, what I believe, two. And the answers, Guatemala or Belize. All right, graduate level. Number four. This peninsula is separated from the mainland of Mexico by the Gulf of California. It's pretty easy, I think. That is the Baja California Peninsula. Nice job. Number five. The name of the largest state, is also the name of a small dog. I knew it because of the dog clue. Yep, Chihuahua. All right, number six. This volcano, the second highest peak in Mexico, is about 43 miles from Mexico City. Oof, I'm gonna mutilate the pronunciation, but it's uh, what is it? Popocatépetl, something like that. Um, I knew it, I just didn't know how to say it. All right, PhD level, three more questions. Number seven, Cancun is a city on the northeast coast of this peninsula. It's easy. Yes, the Yucatan Peninsula. What Mexican state bears the name of a hot sauce? I actually had this on some food last night. The name is Tabasco. And lastly, this brand of Mexican beer is one of the top sellers worldwide. I laugh because, yeah, you're right. It is Corona. Anyway, there you go. Daily trivia of the day. I thought I'd burn a couple minutes there. But back to the real deal here. We're looking at chapter 18. So this is all about gene regulation and how genes are expressed. So if you guys would download the notes for this, chapter 18, gene regulation, it's 18.1 and 18.2 I combined into one. Download those and follow along if you will. We're just going to talk through, and some of this is going to sound a little bit familiar, some of it's going to be new, but that is the key. So if you guys have it downloaded, you can see I do right there. So let us start looking at what we got. All right, there you go. Starting out, chapter 18. If you've read the chapter yet, or if you've looked at it, this picture is actually from the text. This is talking about Drosophila, or fruit flies. They have it color coded, the different DNA genes that are expressed in this Drosophila. You can read about it if you want. It's kind of a cool picture, I thought. But chapter 18, regulation of gene expression. So the first part, 18.1, starts out talking about simple bacteria. So how are they regulated? How is gene expression regulated in bacteria specifically? They use a bunch of examples of like E. coli, which is in our guts, things like that. And the answer is a lot of it is through this idea of either getting transcription to happen or not. Remember, they're pretty simple, so they don't mess with the transcription once it's happened. You don't have like introns and exons and craziness like that. You just have genes that need to be expressed, either turn expression on through transcription or turn it off. So if you look here, this gives you an overview of what's called the trip operon, TRP, um, which is from tryptophan. So E. coli in our guts will use tryptophan as a food source, as a source of um, energy and things like that. So they will operate with this sort of regulation here. And this is a really basic overview, but will have these precursors. They'll create an enzyme, they'll create an enzyme, they'll create an enzyme. And the end product is they are going to make tryptophan. Now, if we have enough in our system already, they don't have to produce it. That'd be a waste of time because they can just use ours. So if that is the case, there are different ways that they will change it. So right here, you can see they regulate it. If we have a lot of this in our system and they don't need to make their own, the presence of tryptophan will then basically mess with the production of these enzymes so that they don't keep making it. How does it do it? Well, it does it by right here regulating the expression of some of the genes that produce the enzymes that are going to do this. So it's just sort of a look at how these pathways, these metabolic pathways are modified and adjusted so that they can maintain control of what they have. So there's two sort of versions. There's a quick response. There's too much of it, shut her down, right? That's kind of a quick response, short term. Long term, if the presence of tryptophan will then, in the long term, modify the gene expression and the production of the mRNA. So we sort of have two different versions. If we look at the next slide, this kind of defines the operon. This was the operon, the term, they talk about it in the book, but it was in 1961. There's some scientists at the Pasteur Institute. They discovered this idea of an operon. And it essentially, if you look at any of them, we'll look at some specific ones, but if you look at any of them, they have sort of these three parts, right? They have part one, the promoter, part two, the operator, and part three, the actual gene that needs to be coded. And this is on the DNA, right? So it's a cluster of these related parts that have essentially an on-off switch, right? So if you look at the simple version, here's your DNA strand. Here is the portion that needs to be coded. So there's the genes. This whole unit is called the operon because it has a promoter region, which is where it gets turned on or off, or where the polymerase attaches. It has the operator, which is where it is turned on or off. And then it has the genes, which will produce the mRNA code right here, which will then, in translation, produce the actual polypeptides. So, real simple. If you want to keep it simple, think operon is the whole thing. Promoter is where the RNA polymerase is going to attach. And then if the operator is triggered, this can then go past and do its job. If it's blocked by a repressor, then this gets stuck and it can't. Simple, basic. All right, so if we look at a few specific examples, this one that I'm looking at right here, this is when you have sort of a little bit more detail. This is with the trip operon, okay? And the trip operon, the TRP operon, there is a sequence. So if you look at what this is, again, bacteria, but if you look at this big strand of DNA right here, quite a distance down, so this is a big gap of junk DNA, if you will, but down a ways, there is going to be this promoter and a second gene. This is the regulatory gene. So this will allow for this operator to be either triggered or not triggered, okay? So what this gene does is it produces a specific repressor. In this case, it is inactive because, right, there's nothing locked onto it. So when tryptophan is absent, the repressor is inactive, which means the RNA will then slide past the operator section and code these genes, which means that this bacteria, this E. coli, will make the enzymes that will make tryptophan. Because there isn't any in our diet, this is not going to mess with this, and it's going to go ahead and make the gene. Okay, so that's one example. The repressor would block if it was activated, it would block. So here are a couple different ideas, and the book will talk about what they call repressible operons or inducible operons. Again, they're very similar. One works where it's always on. That's the repressible one. It's on and you can repress it to shut it off. The inducible is usually off and you have to trigger it to be turned on. So in this case, just like the trip operon I just showed you, this one here is inactive because it doesn't have the co-repressor. In this case, tryptophan itself acts as a co-repressor. So now this gene will be expressed because this isn't being stopped. The RNA is going to make its parts here, and we're going to make our tryptophan producing enzymes. But if tryptophan becomes present, it'll lock on here, right? Here's the co-repressor. In this case, if you want to label it, this would actually be the end or the tryptophan. And in this case, it will actually activate on it. It'll cause this to stop, which means this is then going to be blocked and we get no transcription. So that's a repressible operon. Inducible is just the opposite. In this case, this guy, right, is ready to go do its job. It locks on at the promoter like it's supposed to. The operator has this thing permanently attached. And then to unattach this, you need to have something there that will induce it to let go. In this case, this would be an example, and we'll get to show you one here. But this would be what the lac operon does. So this would be lactose, and lactose would then lock on to this thing, inactivate it, which then allows this to go on and do its job. Okay? Highlighting exactly that what I just said, this just kind of highlights it. And I left all the words in this one. I didn't put them in as skeletal notes, so you guys can kind of add things as we're talking or as you're taking notes out of the readings too. I just left it all there for you this time. But again, repressible ones, they are normally on, right? That is the key, right? Well, let's use a different color. They are normally on, and that is the key. These are anabolic typically. Those are the ones that build organic molecules. So again, if we had to put an example up here, right? This would be the trip operon. That's one example. Okay, so the organic molecule acts as a co-repressor, right? So that's the tryptophan. The operon is turned off when the co-repressor becomes present. So again, here, this is the tryptophan. It inactivates. We do not express the genes. Usually, the gene will be expressed unless tryptophan is present. Then E. coli doesn't have to make it themselves. So that's the repressible. So here it is, very detailed from your text. So this is the picture you've seen. But again, we can look and label. We could even do more here if we wanted to. But if I take a peek at this image, this is our regulatory gene right here. So that's a regulatory. So that creates or makes this repressor. Now, this repressor will stay off unless it is turned on. So this guy will then be able to, our RNA polymerase will be able to go down and do transcription, right? So now you remember this promoter, there's our operator. So promoter, operator, genes, that's the definition of an operon. Okay, so that's not a bad deal. We've seen these before, but just to highlight it and give you a little more specifics. The inducible one, so this is the one that is normally off, right? So this one is normally going to be turned off, and then we have to turn it on to make it work. So this would be catabolic. So again, if you had to come up with an example, this would be, this would be something like the lac operon, which is the one that deals with lactose. So in this case, the repressor is active. It's locked in place right here. So lactose, so this guy would be lactose again. That presence of lactose will then make it release. So this will go down and transcribe. It'll produce the genes that then digest and break down lactose. And again, it makes sense. Why make the genes? The E. coli is not going to waste energy making all these enzymes that are going to break down lactose if there isn't any lactose present. So they will stay turned off until lactose becomes present. That lactose will then take that thing and it'll inactivate the repressor, allowing the genes to now make the enzyme that'll digest lactose. So it's just another example. So you have the trip operon is the one type, the lac operon is the other type, and those are the two sort of versions of the operons. And here again is a highlight of the entire thing. So again, we're defining an operon to be the promoter region right here. So there's the promoter, there's the operator, and then here's the gene. That plus that plus that equals operon. Right, here we have the regulatory portion out in front of the gene. Sometimes it's right next to it, sometimes it's quite a ways away. That's how it is. So it will then lock on normally. So this thing is off. Lactose becomes present. Lactose inactivates the repressor. In other words, it removes it, allowing this to transcribe and produce those, which are going to make the enzymes that digest lactose. So there are two ways to think about it, two ways to look at it. There is both positive and negative control. So when you look at what we've got here, the two ways that they work, fairly straightforward, but negative, the operons are switched off. So keyword here, they are switched off by an active form of a repressor protein. So those are the ones we just looked at. The trip operon and the lac operon, they are turned off by that other thing. The other one, which you haven't looked at yet, and will be on the next slide, is what's called a positive control. So in this one, what happens is the presence of these guys here will actually increase the rates of transcription. So negative control either switches them off by activating the repressors, or positive control, it actually increases the rates. So take a quick look here. This is from your text as well, but this is showing you, this deals with glucose. So in your bodies, or in these bacterial bodies, when glucose isn't around, so there, again, this could be like an E. coli that wants to digest and use glucose as a food source, right? If there isn't any glucose around, it's becoming scarce. So what happens is this molecule called CAMP, cyclic AMP, CAMP will accumulate when glucose isn't around, okay? But what'll happen is that CAMP right here will bind to this inactive CAP, which is a catabolite activator protein. So those two will combine to make this unit, and it'll activate it. So here it is activated. And basically what happens is that activated unit will lock onto the promoter. Now, it doesn't try to block it. It doesn't try to make things go that way. What it actually does is when this thing is present, it actually makes it easier for the RNA polymerase to bind. Typically, there isn't a great affinity for this RNA polymerase to find that promoter. But this is basically like somebody standing there shouting, hey, right here, right here. And it actually increases the affinity of this to bond to this, which in turn will increase the rate of transcription. So it's basically a way of speeding the process up, okay? So those are the different types of negative versus positive control. In this way, when it locks on, it actually speeds it up. In negative control, when this thing is in place, it's in front and it would actually block this. So that would be negative control. This is positive control. So that's 18.1. And again, that's probably 6-7 pages if you want to read through them. There's a lot of technical terms and stuff in there, but again, we're kind of looking at the idea of what happens when you mess with different portions of this, okay? That was prokaryotes. Now let's look at us. Let's look at eukaryotic cells. How is it regulated here? It's not just turning it on or off. There's a little more to it, right? So we have, excuse me, a whole lot more stages that are going to happen here, okay? So what we need to look at then is the overview of why. So typically human cells, you only have about 20% of the genes being expressed at any given time. So again, and it relates to the different types of cells. So all of your cells, and again, there's one exception, it's the immune system cells are a little different, but pretty much all of your cells are going to have the entire human genome in each and every one of them. But if you are a skin cell, you have a much different need than you do if you're a nerve cell. So you're not going to code, you're not going to go through and transcribe all of that because it'd be wasted, because you don't need all of those parts to operate as a skin cell, which you might need as a nerve cell. So you have to turn on different parts and pieces of them. Now, obviously, one of the ways you do that is through, and this is at the end of this chapter, but it talks about how introns and exons come into play. And we've already talked about that, so I'm going to leave that part up to you guys to just review that again. But that's one, turning on and off by using different introns and exons. That's one way. But there are other ways that there can be differential gene expression other than that. So that's what we're going to take a look at here real quick. It can be regulated at all different steps in this. So if you look at that image, there you can see here, there is even before we get to the DNA, there is some modification that can happen right there. All right. Imagine that if I had a message on a piece of paper and I balled up that piece of paper and I said, here, read the message, but you can't unball it. Well, you can't read it, right? So your chromatin will stay balled up like that, which makes it a lot harder to get the DNA code out of it. So if you can somehow unwrinkle that paper, just like you have to unwrinkle that DNA, that's one way to control expression. If you don't allow that to happen, you're not going to express it. So that could be up in here. You could have transcription. Well, you have to turn it on, right? Maybe the whole scorpion tail thing. So we'll talk about that and how you turn it on. So there could be some stuff there. You could talk about the RNA processing, which is the introns and exons, and how that can change the code. So we've already got 1 there, we got 2 there, we got 3 there. Once the code is out in translation, we could have some degradation of the mRNA. So there's enzymes breaking away that mRNA to break it down before something can happen. So that could cause some problems. We have the different folding things that happen after the protein has been made. And then we have the moving it to where it goes to. And if you block any of those, you kind of mess with the regulation. So it can happen at all these different parts. This is actually be a very good sort of essay question to ask about what would happen if, right? And you'd have to explain different parts of the process. So anyway, here's the one I was talking about with the paper, right? So if you had that chromatin structure and it's really tightly bound, it's going to be less accessible for that RNA polymerase to get in there and actually transcribe it. So there's 2 ways that this kind of works. When you look at the images I have here, these are, again, from your text, but you have what's called DNA methylation. And on the next slide, I'll show you what that looks like. But DNA methylation means that you have methyl groups that are added to the DNA, which even pack it even tighter, right? So when you add that methyl group, it cranks it even tighter, which decreases the rate of transcription. So that's one way to slow it down. Then the other one is with histones, and this is acetylation. So in this case, you're actually adding, instead of methyl groups, you're adding acetyl groups to the histone. Remember, DNA is, so DNA, we always think of it like this, but it's actually wrapped around these protein histones, and that's what causes it to become all chromatin like that. So if we sort of loosely wrap it around the histones instead of super tight, then RNA can get in there, RNA polymerase can get in there and actually do the transcription. So this way right here, so methylation would slow it, and this would speed it up. Okay. So here is the picture that I was mentioning before, right? If you look at it, this version, right? Let me go back to this version right here, super bunched up, right? You can't get an RNA polymerase in there to actually do anything. It's just too balled up. This is my ball of paper. Where in here, it's been acetylated, so it's actually spreading. This is the histone, that's a histone, that's a histone. So it's actually spacing those histones out a little better. That way we can get in here and we can actually get to it, right? And do the transcription, right? So another great way to look at it. Okay. Okay, this is kind of cool. This stuff, this epigenetic inheritance is something that's pretty new. Our book touches on it, but there's a lot online you can find about this. It talks about modifications of not the genes themselves, right, that you inherit from your parents, but it talks about modifications on the chromatin that can be passed on to future generations. So they call it epigenetic inheritance. It's not the actual code, but it's things that can happen to the coiling, right? DNA mutations, you can't change those. Once they happen, you can't go back like when your child inherits them. You can't go fix those. They're inherited. But these, you can actually do demethylation of the DNA. You can actually change them back. So in this, I'm going to have you guys stop this video, and there's a little clip I want you to go look in a second here. But it talks about identical twins and how 2 identical twins might not be identical because of this, because of some of the epigenetic changes. So, and there's some examples here about gene silencing, the methylation or the acetylation, some X chromosome inactivation, some of the stuff that we don't delve into a whole lot in the text. But I'm going to have you guys right here hit pause And if you will, there's a link you can go to, this epigenome at a glance. It's about a 1.5-minute video from the Genetic Science Learning Center. Hit pause, open this one up, watch it, and then come on back and we'll talk about it. All right, you're back. So hopefully you found that interesting. I thought it was kind of cool. I know it's short, but if you go to that page where that was listed, you can kind of search around in that genetic page, and there's some cool stuff about it. So use some of those resources if you have time. Take a look at it. What I want you to look at right here, again, is this is a zoomed in from what I showed you earlier. But this talks about, so we already talked about this, how you can mess with that chromatin and make it harder to read, harder to do transcription, or easier depending on if you unpack it or leave it packed. But now let's get into the actual DNA. So here's our DNA. Now we got to transcribe it. Okay, how do we get through and do that? Well, this goes back, again, that video, a lot of you remember that little scorpion tail, and I'll have a link to it in Schoology, so if you want to go double check it, it'll be there. But now we have ways to either turn on or turn off specific transcription factors to make the process happen, right? So we have what we call activators to make it happen, or repressors to slow it down or turn it off. And basically that's what we're looking at here. So again, if I look at this image I got here for you, this is what you guys have sort of seen before. So again, we have essentially our operon here. We have our promoter region, we have our operator region, and then we have our gene. So there is our operon, right? And again, this starts to bring back things you might remember. The specific promoter region, a part of it is called a Tata box in eukaryotic cells. So that Tata box is right here. Then there's the operator, and then there's the genes. So a distance out from it, and it could be quite a ways, could be thousands of bases, it could be a long distance away, it could be fairly close. But way back here, there is a controlling section. So there is a what are called enhancer regions. And when an activator binds to that enhancer region, it then allows for this, the scorpion tail, so this DNA to bend over and then touch right here. And these activators will then bind with all of these different parts, these mediator proteins, we call them basal factors, these general transcription factors. All of those will clump together to make this transcription initiation complex right here. And then that will allow the RNA to grab on and then to progress and do its thing. So we got to have this whole sequence of stuff to happen. If we have, if we're missing these, it's not going to work. If we're missing some of the transcription factors, it's not going to work. So there's a lot to sort of activate and make this thing happen. That's it. That's what I got for you. I know it's a lot. I'm at like 20 minutes here, 25 minutes. But that's chapters 18.1 and 18.2. So this is in a little more detail on there. But if you guys want to, I have a couple video links I'll put in Schoology so that you guys can kind of look at some of the examples, some of the HHMI animations and some of that. But that is sort of our basic regulation. And then tomorrow, I'm going to have you guys look at 18.3, 18.4, 18.5, and we'll kind of talk about some of those aspects as well. That's it for today, guys. Hey, I heard a good joke today. See if you guys like this. My wife said it was a dad joke, but here it is. Do you know the difference between people in Dubai and people in Abu Dhabi? Well, the difference is people in Dubai, they don't like the Flintstones, but people in Abu Dhabi do. No. Anyway, have a good day. Stay safe. Get outside and enjoy the weather while it's nice. And stay distance. Wear your masks. Let's flatten this curve. Have a good one. We'll see you later.